Hello, everybody. So far, you've been studying what's called Mendelian inheritance or complete dominance, where one gene is completely dominant over the other. Therefore, you call it the dominant gene, and then the one that it covers up is the recessive gene. Well, it turns out not all inheritance follows what are called the Mendelian rules or the complete dominance rules. There are other forms of inheritance out there, and that's what we'll talk about next, and then we'll learn a new way to do those problems. These two new types of inheritance are called codominance and incomplete dominance. Both situations are very similar, but there is a slight difference between the two that we'll talk about. But we handle their problems with the Punnett squares very similarly. All right, so here's a new situation. Before, what would happen if we crossed a red flower who is pure or homozygous for its trait of red flowers with a white flower who is also pure or homozygous for its color, we would expect to get one or the other, either red or white, under the old rules of Mendelian or complete dominance. But in this case, when we cross a homozygous red with a homozygous white, we don't get red or white, we actually get an intermediate pink. In fact, 100% of the offspring are pink in this cross. So this is called incomplete dominance. So it's the second one on here. Incomplete dominance is when you get a blend of the traits. The traits are blended. So red and white makes pink, or maybe like a purple and a white would make kind of a lavender. Here's some other situations. By the way, these are called snapdragon flowers, and this inheritance is common with these flowers. Cross the red with the white and you get pink. These are called permanent chickens. They come black and white, and when you cross a black and a white chicken, you actually get gray. A mixture of the two. And then here's another one, a type of shorthorn cow. Here we have the white bull on one side, and we have, this is, yeah, I know it's kind of a burnt orange, but they call it red. So cross a red with a white cow, and we get what's called roan, which is an intermediate blend of the two, neither red nor white, but a mixture of the two. And then these are 4 o'clock flowers. I used to actually plant these when I was a kid. You have just like the snapdragons, red flowers and white flowers. And then when you cross them, you don't get red or white, you actually get pink. Again, this is called incomplete dominance. It can actually happen in other animals as well, but it is quite rare. It's more common for the complete dominant situations to happen. But again, there are some of these situations with incomplete dominance that does happen. All right, now I've switched to a board that I can write on, and hopefully I can do a good enough job writing on the computer, so you'll have to excuse me. So what I'm going to do is do some sample incomplete dominance problems. So these are R's. So what I'm going to use is 2 capital R's to indicate a red flower. I'm using 2 capitals here, 2 separate capitals actually, because the white flowers are going to be WW. So you'll notice a difference already with these problems, because before we would just pick one letter and the dominant allele would be the capital letter and the recessive would get the lowercase, such as big R, little r, or big W, little w. And actually some people do those problems that way, but then they have to remember a little detail with the heterozygotes. So the way I prefer to do it is, because neither allele is dominant over the other, neither one covers the other one, then I feel they both deserve capital letters. So I'm going to use capital R for the reds and capital W's for the whites. They get their own capitals.
So here's what the Punnett square looks like if I cross a red flower with a white flower. To be red, there's only one way. You have to have two alleles for red. So I'm using capital R's, putting them in the proper spot on the squares. It doesn't matter if I do top or side. And let's cross this red flower with a white flower. The only way to be white in this incomplete dominant situation is to have two W's. All right, now let's fill in the boxes. It doesn't matter if I write the R first or the W first, but I would like to keep it consistent in each box. So I'm going to pick the letter I want to write first. Each box, again, you bring the big R down, big W over, and write them in the box. I'm going to do R's first, W second. So every box has an RW. So what happens when you have heterozygotes, RW? That's when you get pink. So if I were to cross a red flower with a white flower, 100% of the offspring would have the phenotype pink. And 100% of the offspring would have the genotype RW. Right? So again, here were my homozygotes who were red. Their genotype, big R, big R. My homozygotes who were white, genotype, big W, cross them. And I get genotype RW. And phenotype is pink. Again, this is the situation for incomplete dominance. I hope you are following along and writing some notes on how to do these problems, because you will be getting problems to do. Right, so this is incomplete dominance, and again, it's hard for me to write on the computer here, but incomplete dominance, and this is when you get a blend of the traits in your heterozygotes. Again, homozygous is when you have two of the same letters, like big R, or big W. And then heterozygous when you have one of each, R and W. So incomplete dominance, you get a blend. Now we can do some other variations on the same problem. All right, I'll clear that and let's try another one. Let's say that I take a pink flower. So again, pink, we know in this situation of incomplete dominance that to be pink, your genotype must be big R, big W. Let's take a pink flower and cross it with a red flower. Again, to be red, then the only way is you must be homozygous. You carry two big R's. Right, so let's make our Punnett square. It doesn't matter which one I put top and bottom. Let's put the pink one up here, RW. Let's put the red one here. And then let's fill in the boxes. Right, we see in the first box, 1 out of the 4 boxes is red. Heterozygote here. 1 out of the 4 is pink. Oh, now we've got another red one. So 2 out of the 4 boxes is red. 2 out of the 4 pink. So now I'm going to reduce that to 1/2 or 50%. So I would expect half of the offspring with this cross to be red. That would be the same genotype, big R, big R. And then I would expect 1/2 to be pink. Trying to write with my mouse here. So RW. All right. Let's try one of the chicken problems. The erminate chickens. They come in black. The only way to be black is you must have 2 big B's.
They also come in white because we know through different experiments, we know this is a case of incomplete dominance. White must be two big W's. Okay. And then when you mix those two, if I did a cross of BB with WW, then from our other experiments, we know that we would get BW or WB, but I'll be consistent and do BW. That's going to be gray. A mixture of those two. So incomplete dominance, that's the blending effect. Let's say that I mix two gray chickens. Cross two gray chickens. What will I get? Right? There's gray chicken mom, gray chicken dad. Let's put them in the squares. Again, be consistent. If you're going to write B's first, write B's first. You're going to write W's first, then write W's first all the time. Let's bring our W's here. There. All right, so what do I expect for my offspring? Well, 1 out of the 4 boxes is black. Or I could say 25%, so 1/4 or 25% black. Notice that 2 out of the 4 squares, or half of them, or 50%, are gray. So right here, all right. And then 1 out of the 4 squares, or 25%, are white. As I've mentioned before, there's different ways that I've seen these problems done. Some people use the old notation using like a big B. They pick one of these colors, black or white, to have one letter, like a big B, and then they use the lowercase for the white, little r. But when you do that, then you have to remember that those two alleles are dominant together and you're going to get a blend. And so then big B, little b would be gray. Again, it can be confusing if you do it that way, but I've seen people do the problems that way. But again, I prefer, since neither allele is dominant over the other, I prefer that each allele gets their deserved capital letter. And then your heterozygotes are easy to see, W, and you know that you're going to get this different situation, this blending situation where they are gray. Right? So there's those problems. If we were doing the problems with the cows that I talked about here, I might choose big W for a white cow, big R for the red cow, and then my roan intermediate or blended form is big R, big W, a lot like the red and white flowers, except you don't get a pink cow. Wouldn't that be cool if you did? You get the roan cow. So again, this is called roan. Here is a picture of Punnett square there using the R's, W's for the 4 o'clocks. Here's a picture where someone has chosen to use the original letters like big R for red, little r for white, and then the pink flowers are big R, little r. But again, then you've got to remember that both alleles are actually incompletely dominant, and you're going to get heterozygotes that are totally different color, totally unexpected pink. Right? So the parent generation, we call the F1 generation. Here, the second or the first generation of kids. F2 generation, the second generation of kids. Some terminology there. All right, let's take a look at the next situation that we have mentioned, codominance. Remember, there's a prefix on here. Co means together. So it kind of helps you remember what this means. So while incomplete dominance was when you blend the traits, like red and white making pink, codominance is when you can actually see
both traits separately. So the organism will show both traits. And the best way to show this one is through the human ABO blood types. So with the ABO blood types, we have a few things going on here that these are really kind of interesting. Is that O is still recessive. So that O falls under the old rules with complete dominance, that A and B are actually completely dominant over the O, and O is recessive. But then we've got multiple alleles. We've got more than one choice here, not just like red or white. We've got more than one choice. We've got A, B, and O. That's called multiple alleles. And it turns out A and B are both dominant over O. So that part of the situation is still the old rules with complete dominance. But the new rule here with codominance is that A and B are both dominant together. So anybody who has AB alleles will show both the A and B blood types. All right, so what are the blood types? Well, it turns out that these blood types signify a protein on the cell membrane of the red blood cells. So here is what an A red blood cell looks like. It's got a little protein on the outside. We'll call it the A protein. And then here's the B red blood cell. It's got the B protein on the outside. Looks a little different than the A protein. And of course, our immune systems, if you put the wrong kind into someone, the immune system would notice these proteins, and it does. So we have to be very sure when doing transfusions that we're getting the right type in the person. Somebody who gets AB blood will show both. Notice this is not a blend. It's actually both proteins showing up on the red blood cells separately, individually. We can actually see them. Little round ones, there's an A protein and there's a B protein, the little spiky ones. And then there's the O situation. The O doesn't have either protein. So you have to be very careful if you transfuse A or B or AB into someone who has type O blood. Those are foreign proteins that these cells have on the outside of their cell membrane. And O person's immune system is going to notice those proteins and then attack them, and then it could kill you. So we say that O is a universal donor because it doesn't have either of those proteins, what we call antigens, on the outside. So O can give to A, B, and AB without problems, but not vice versa. And AB is the universal acceptor, because AB can accept from O, from AB, from A, and B, because there's no weird proteins on the outside that will make the immune system attack. All right, so how are we going to do these problems? Let's take a look at that next. All right, we're out on the Jamboard again. And so here is type A blood. So for type A blood, what I like to do is do this, that type A blood can either have two A alleles, or it can have an A allele and an O allele. Remember, O is recessive, so I made a little O there. Type B blood, you can have two B alleles, or you can have a B, which is dominant over the O. All right, B will cover up the O. Then AB is pretty self-explanatory. That's our codominant situation, where you have both of those alleles. Right, so that's all I have to write there for AB. That's the only choice. You get one of those from mom, one of those from dad. And then your red blood cell displays both proteins on the outside. For O, there's only one choice.
because it's recessive and anything else would cover it up, it must be homozygous. It must have 2 copies of that recessive allele. Right? And so now I can do some problems. So let's say that I cross somebody who has type AB blood, so that would look like this, with someone who has type O blood. So that would look like that. And I'm going to follow the old rules again by writing the dominant allele first. So that means bring the A down, bring the O over, write the dominant one first. Same thing with the B. So what are the results that I will expect in the offspring? Well, it looks like 2 out of the 4 or half or 50% of the squares will have type A blood. But they're going to be heterozygous. They're going to carry that recessive O allele. It'll just be covered up. But their red blood cells will display the A protein. And then half, or 50%, will be type B blood. Again, they'll be heterozygous and carry that O allele, but since it's recessive, it will be covered up. Let's try another one. This is a fun one. Let's say that we have 2 parents and they have a kid with every blood type. How is that possible? All right, how is it possible? Well, let's give them at least this. We know that's going to give them AB, right? So how can we get a kid with A type blood, B, and O type blood? Well, we're going to have to make each parent heterozygous, right? Because now we've got AB here. Oops, we've got B, we've got A, and we've got O. So it really truly is possible for parents who have different blood types A and B to have a kid of every blood type. Each time they have a child, each of these is a 1/4 chance or a 25% chance that they'll have an AB kid, 25% chance they'll have a B kid, 25% chance for an A kid, and 25% chance for the O kid. Right, so this is how I do those problems. It's just that in blood type problems, you have to remember that there's the situation of multiple alleles, that you've got A and B showing codominance. They're dominant together, and then both alleles, the phenotype will show if they get the heterozygous condition. And then there's the recessive condition with the type O blood. All right, then I want to show you again, as I mentioned in the incomplete dominance, that there's different ways to display this. And different books and different people, different teachers are going to use different methods, unfortunately. The way I do it, I feel that that's the easiest, but some individuals use what are called superscripts. And so they use I as their symbol for blood type. It actually stands for the word immunoglobulin, which is a description of the proteins that are on the outside of the red blood cell. So they use capital I and then the superscript A. So someone who has type A blood could be this, or, and superscripts are kind of hard here with writing on the computer. There. So it's 2 capital A's or capital A and then O. So I've seen them do that, or they've also used a little I for their O. So I've seen them do that as well. And for B blood, capital B, so B blood, or again, capital B or an O here, or they might use a little I for their type O blood, which is recessive.
Then of course, if you have type AB blood, then you get one of each. B. And if you have type O blood, I've seen them do this little I, little I. Right? So the problems would look the same. It's just that you've got that extra step of putting the I's in there and doing superscripts. In our next type of problem that we're going to do this week, we'll do what are called sex-linked traits. We actually will use superscripts, but maybe won't be as complex as we see with blood types. But again, I feel it's a lot easier to use this method. It's the method I learned when I was in high school and in college. All right, so again, this is complete dominance, where you show two alleles who are both dominant together, and you see both phenotypes in the offspring. And then there was the blending with incomplete dominance, right? So now you're going to get some problems of your own to try. And I'm going to try to get a copy of this Jamboard and the video and try to put that on your Google Classrooms if you need to go back to it for reference, right? Good luck on the problems, and let me know if you have any difficulties.